Well, thank you for the introduction. And I love it when you said the whole journey because that to me means I'm old. And it's correct. I'm old in that I recently retired. So my conflict is actually pretty clean. It's with the company that I helped started, Cook Mouse Site and Lapella. All right. So I'm talking about crossing the different valleys of death that we all cross. I'm sure everyone on this call is probably crossing a valley, getting that PhD finish, getting that postdoc, finding that first academic job, getting your first grant, getting tenure. So I'm talking about a little bit different a valley of death because after you accomplish those, what's in your future? Well, there's more valley of death. If you have a great idea, how do you move it from the preclinical to the clinical? If it's really a good idea, you can't stay within the university. You have to get it out of the university and into the industry. And then the biggest valley of death is getting the FDA to approve you to do the first patient clinical trial and then subsequently on to hopefully toward approval. So the journey ahead of you is many more valleys of death, but you could do it since we can learn on the job. Hmm. And Christine introduced me, but I just want to reframe that. I was very well trained, but I was trained as a clinician. And it was when I was at a Thomas Jefferson University, I decided that being an MD, I want to do more research. So I moved over to the University of Pittsburgh to work with William Chat DeGroat and thereby starting off the preclinical research toward development. And in order to do that, I had to move to Corewell Health, where they're at Beaumont, where they really have a lot of expertise on the clinical translation and clinical trial. And on the bottom, these are the grants I have been the PI or program director on, from R01 to PO1 to physician scientists, K Awards, and the PhD T32. And I'll talk a little bit specifically about the STTR, SBIR that I've received, the R23 and R24. I think these are terrific sources of funding if you're heading down the translational path. I've also been involved with the DoD, foundation industry, and philanthropy from my patients. But as you cross these values, because everything research is driven by money, you need money to do anything. And once you get to toward out of the university, then it actually gets really about money because you really got to be talking about investor, venture capitalists, bank, and financial institution. And they are much tougher than NIH or a paper reviewer. So I'll talk about 7 things, and I'll use radiation cystitis as the example. But if you see the bottom, this is not a talk about the science. It's about the lessons learned, many of them is through mistake I've made along the journey from what is an unmet need. How do you get the right model to show it, but not stay preclinical, but move it into the industry? I'll show you why I chose such a generic drug, tacrolimus, for development rather than a novel, exciting de novo protein. I'll talk about the key lessons I've learned from the Valley of Death moving from the academic to industry. I'll have a slide on the SBIR, a slide on the orphan drug, and a slide on the 505(b)(2) reposition pathway. That's really good way for the FDA. And finally, how you got to get involved with patient advocacy, different stakeholders, because the FDA really look at stakeholders' involvement in helping you move it into clinical and move it forward. So hemorrhagic cystitis can be due to radiation or chemotherapy that damages the bladder and causes bleeding. I'm not going to read every word on the slide, but I deliberately put it up as this is the type of slide, if you're going to go in toward investors, sort of what they expect in one or two simple slide, not go into the detail, but summarize what's the disease and potential opportunity that they should consider. But there is unmet need because there's over 15 million Americans who are cancer survivor. So the example that what the market we're going after is cancer survivor disease. And it could be due to radiation for prostate, uterine, rectal, or chemo for systemic treatment that could cyclophosphamide that could damage the bladder. The number I'm showing also is like, well, it's not that big. I mean, out of what, 700,000 prostate cancers, that only 42,000 has radiation hemorrhagic cystitis. Well, that's too small for my drug. But a number below 200,000 prevalence is actually excellent and required to get an orphan designation. So a precise number from all available literature through medical record data, Medicare data is a must for venture capitalists and actually for the FDA. So we talked about the hemorrhagic radiation cystitis, and only about 5 to 10% of patients who gets pelvic radiation will get it. But the opportunity is there because there's no FDA approved drug for it. So currently people put alum, silver nitrate, formalin in bladder installation, or you have to put somebody into a hyperbaric chamber, 2.5 to 3 atmosphere. But that takes 30 to 40 sessions over a 3 months period. So if you're bleeding this weekend, it's not going to solve the problem now. So lack of approved therapy, a orphan population really opens up the regulatory pathway, and the investors in the biotech space knows that also. So we went down these paths, and I'll share it to you. But the mistake I've made is it took me a couple years before I really figure out a focused approach in a disease that there's no competition. And that's although rare, it's very expensive and it's severe with 4% mortality is a good way to go. But the lapella, although it sounds like I know what I'm talking about, it's really lesson learned the hard way. And before I started lapella, I had the idea uh, over 20, 25 years ago of using a muscle-derived stem cell as an injection therapy for female stress urinary incontinence. This is an alternative to the sling or a bulking agent injection. So I developed this at the University of Pittsburgh. I seeked out, I'm a physician, so I seeked out a cell biologist, Johnny Uar, who's doing skeletal muscle regeneration for Duchenne muscular dystrophy. I talked him into working with me. We got a $50,000 seed grant. This was during the nascent period of stem cell development, so I recognized the opportunity, and we got our patent submitted relatively soon, and that led to a R01 on the first try. And the opportunity was just right, and a large device company, Cook, licensed it from the University of Pittsburgh. They own it, and I'm just been a consultant with them for the entire duration. But now this is in finishing up phase 3 trial, and the final phase 3 pivotal results will come out sometime this summer. So much of what I've done in Lapella, the startup company, I've learned from the lessons from a licensing deal with Cook MyoSite. What I really like to share with this group is, although I'm doing the talking, I'm definitely I feel like definitely you can't do it alone. To me, a mentor, colleague, and pupil is absolutely required for me to be successful with them together. I mean, DeGroote is the one that taught me science. Naoki Yoshimura is my close friend and partner. And I found one of the top liposome expert in the world, Li Huang, who happened to be in the same department as Naoki and DeGroote. And I talked him into developing liposome to put in the bladder as a platform drug delivery. He thought enough of the idea that he assigned his PhD candidate, Pradeep Tyagi, who many of you know now is an associate professor, to be his PhD thesis, and that led to an R01 and his recruitment to the urology department. So I work with every one of these people today, and without, and my pupil clearly knows more than I do on the topic. The second, how much clinical do you need? And I would say you need much less than you think, but it has to reproduce what the clinical condition mimic and what the venture capitalist would associate. So there's a lot of way to radiate cells and thing, but if people's going to invest money on an idea for radiation cystitis, they really want to see, well, can you create an animal model that shows this and that your drug works? So at Beaumont Corewell, we have a small animal, the SARP machine, that's mainly used for brain cancer and lung cancer, but it's very much like the human radiation for a man with prostate cancer. So we just say, let's go ahead and radiate the bladder, and we talk the SARP team into letting us using the machine. So on the left is a CT that you can give in contrast. You can draw out the bladder and you can dose the radiation to the bladder. And on the right side, you can notice the change. This is mimic the human, and the liposomal tacrolimus had an effect. And that was enough to move forward to satisfy the FDA and move forward. So at Corewell Beaumont, we had the similar circle of a relationship. Our mentor, Ananias Diokno, colleague, Chairman Ken Peters. Jason Heffron is an oncologist, but he has the patient for a radiation oncologist, and he did the trial. And mostly his pupil, Bernadette Zwan, that many of you know. She finished her oncology PhD at the University of Pittsburgh, excuse me, University of Michigan. She wanted to do more translational research, so we were fortunate enough to recruit her, and she just completely ran with the radiation cystitis, the disease and model. The first one at Beaumont Corewell to get a Urology Care Foundation grant, the first one to get a K award, a KO1, and moving this onto an R01 after the K award. So she knows more about the disease than I do, but it takes a team to bring it from the model all the way into the clinical trial. And I'll give you a snippet of the trial that Jason Heffron did. So this is where we get interesting. What do you give? Is it gene therapy, knockout? We're using an old drug, tacrolimus. It's been generic for 20 years. It's probably the best immunosuppressant, but it has a terrible systemic side effects because if the blood level for tacro is high enough, it causes small vessel constrictions and has significant renal toxicity. But if we formulate with liposome and tacro is highly lipophilic, we can achieve very high concentration in the bladder urothelium, suburothelium area, while minimize significantly lower systemic absorption. And there's a very simple whole blood test for tacro, so you can check if it gets into the blood system or not. On the right side, how do I mean, I'm going to keep saying it. Everybody's been told you got to have patents. Well, so what do you do with tacrolimus? It's 20 years old. It's patent, it's 30 years old. But the two things you really do need are, if you could do it, is the second one is a method of use. Putting liposome tacrolimus is novel, but that's not as strong as the composition. So we have some degree of protection on the liposome, specific liposome, and the tacrolimus, and it needs to be filed before any publication. So throughout the years, through Cook and Lapella, became very obvious that you just gotta have some sort of patent. It doesn't have to be the world's greatest patent, but you gotta do it before anything gets out there. And because for people to look at you and invest in you, and even the SBIR, STTR grant, they want to see that you have patent to be able to move forward. So I found it very useful to work with. I found a good relationship with the director of tech transfer at the University of Pittsburgh back in those days, and it was very useful moving it forward and getting it filed on time. So, and get the composition of matter and field of use. you really be ideally, the composition's the better, but having both would be preferred. But even with all my intention, I almost totally screwed up on my cook mouse ideal. So I knew we had something interesting. So we filed the patent, we submitted the R01, we submitted the paper, and an abstract to present at the AUA. But the patent was submitted before that during that time, but it was certainly become noticeable. But 2 years after the patent was issued, we actually got challenged by a European company stating that we disclosed the patent because of the AUA abstract. We submitted the patent sometime in April, and as you know, the AUA is in May, and they challenged it, and it took about a 2 years battle back and forth, and we had to subpoena the librarian at the University of Pittsburgh Medical School's library to testify that the abstract was not disclosed until 2 weeks before the AUA meeting. So therefore, our patent was in the not in the public domain because it wasn't on the library shelf 2 weeks before where we submitted the patent. But it got down to that nitty-gritty on when an abstract was presented. I just want to give you an idea of what some patent looks like versus the title of your paper. Just the top one is the stem cell, and the bottom one is liposome. If you just look at the title of these two things, you say that's ridiculous. It's like, Mike, you're working on like cell therapy for stress incontinence, but your patent is called soft tissue bone augmentation of yada yada yada. What is that? And the bottom one's even crazier. You're doing liposomal tacrolimus, but the name of your patent is Method and Treatment for Bladder Dysfunction. I mean, where do you go with that? But then in reality, when we submitted the cell therapy, we were more interested, my colleagues was more interested in as a potential a differentiation into the cartilaginous and bone lineage. So it was more interested in orthopedic than urology, thereby why it was listed like that. But you notice the word composition and treatment in the title. And in the bottom, the broader the patent, the broader the name, the better. Although really our examples, liposomal tacrolimus, this is our title for that patent. So tacrolimus, it, it's a potent inflammatory agent, but it has a severe vasoconstrictive effect. That's why you got to be so careful in its use. But it's an old drug, so we took advantage of it by the 505(b)(2) pathway, and I'll show you a slide on that in a minute. And given that the disease is rare, we pursued it for the orphan designation, and I'll have a specific slide on that in a minute too. And this can help you with a multiple accelerated pathway at the FDA. You can get to clinic faster. But before that, I'd like to talk to you just about manufacture. So the drugs we use in research in a rat or mice experiment is not probably not GMP, and cannot be used during the clinical trials and certainly not to a regulatory approval. So from day one, before you leave the university, you got to think about how we're going to make what I'm interested in for human use for clinical trial. Because you gotta have be your GM. It's got to be GMP graded. So either you can figure out a way to manufacture at a GMP facility and have your CMC all figured out, or you have to have a CMO out there that you can partner with that gonna make it for you at a human treatment at the GMP standard. So Lapella, the ingredients for liposomal tacrolimus essentially is just those two things. So it's a very simple lyophilization fill finish product. So we're able to use what you see this really a portable like a tent. It's a uh, portable lab, but it goes down to, with the HEPA filter on the roof, it goes to an N of 100. So it's much cleaner than an OR, which is like 1 in 10,000, I believe. So we're able to make the product at GMP because of the simplicity of the ingredients And we can purchase the tacrolimus because it's a 505(b)(2) and mix it with the lipid. So it's a very simple manufacture. But if you have complexity, you got to get your CMO all ready to go. So crossing the valley of death, you have a academic is good enough for proof of concept. But if you're going to want to help patients, you have to figure out a way to get it out of the university through either a license or a startup. And for an industry to license or partner to start up a company, they got to know that you can manufacture your stuff at GMP and that you can be FDA for an IND, investigational new drug. You're ready to go because nobody's going to want to pay you, me, to do 5 years of excellent preclinical research at high impact journals. So the real value and the only value is getting something into human. So with the lapella, with the radiation cystitis, we found it since I had the experience with the licensing, and I had friends that had the experience in biotech startup, being the CEO and the manufacturer. We have the know-how, so we're able to form a startup that licensed the patent from the University of Pittsburgh into the startup company. So the lessons I've learned is I got to surround myself with people that knows what they're doing, both in the academic and also in the startup side, because the ability to speak the lingos to the investors and the incredible amount of legal works because when you start taking people's money, they're going to hold you really accountable for it. It's really, really difficult. And applying to the FDA for a human trial is harder than getting an IRB for a clinic to do a clinical trial within a university. So these things has to occur in parallel. You got to have your GMP, your CMC all ready to go, or your partner, because any of these things will kill you, and you just essentially raising your money each time to get to the next level. And the faster you can get to the FDA and start having a conversation, getting something in writing from the FDA will really help your ability to raise money because you'll de-risk what you're doing on with the investor. Something in between, if there's a company that's small, that's interested, and you're in the university, I would highly recommend you can think about the ART 43 and 44, the STTR, SBIR, because there's a small 6 months phase one, 300,000 something. The phase two, which I've never had a phase one that didn't get the phase two. So together it's about $3 million. And the phase two is usually for 2, 3 years. So it's really a very reasonable amount of money that for an SBIR is split 50-50 it can be between academic and the company, and that can be used for manufacture. Or the STTR, more of the money can go to the university side. But I can say that the NIH have more money allocated for the SBIR than STTR. And just as a reviewers, the SBIR is more money, easier to get, and I think is better than STTR because people won't be as skeptical that this is just not a shell company. Somehow you're just trying to get the SBTTR money to fund your preclinical research, that you really don't have somebody at a company that's credible that can move this forward into and through clinical trial. Too many people go down the pathway saying, I can try to fake him out. This is really, but the reviewers all have the startup experience. They can see right through it that if there's not a legit CEO or a physical space that can run with an idea, because they shouldn't be a front just to fund your research. Orphan drug, a disease with prevalence of less than 200,000 in the US. It doesn't matter any other country. And it could be more in the future, but 200,000 at the time you apply can qualify for the disease. Radiation cystitis, we have the prevalence data. It's about 70,000, and there's lack of approved therapy. and therefore we received the orphan designation. Well, why is that important? Well, it's important because we have the designation for tacrolimus for radiation cystitis. So if it gets approved for 7 years, there's market exclusivities. Pfizer just can't come in with anything tacrolimus to treat radiation cystitis. There's a tax credit for your costs on trial, reduced FDA filing fee, and they'll work with you a little bit more to give you some guidance. They're still not nice, but nicer. So, sounds like I know what I'm talking about, and I do now, but, I wish I did it sooner. Again, it took me too long before I figured out that, going to orphan, getting the FDA accelerated pathway through breakthrough and fast track, it's possible, but you gotta do it early, and they save a lot of time and they add a lot of value to potential investors. 505(b)(2). Well, tacrolimus is an old drug. Why is that not a bad thing? Well, it's not a bad thing when we could think of a way to get through the patent, the liposomal formulation, to be able to apply for the patent. But what's good about it, because it's a known entity, it means the FDA's already approved several tacrolimus drugs, both IV and oral. So you can reference those toxicology studies. So the FDA really, it's not just what you've done preclinical in toxicology, but what every company's done for tacrolimus for any indication can be considered. So it really could save millions of dollars and years off. And that's one of the reasons why the FDA let us move right to a phase 2A trial rather than just doing a phase 1 trial. So again, that there is a reason sometime to reposition an old drug. And you can get to and you can get to clinical trial faster too. So because it's an old known drug and it's an orphan indication, our first clinical trial was really one patient. It was an expanded, compassionate use, expanded access. And by getting an IRB and FDA approval to do one compassionate use patient, we had first in human treatment experience. And that data led to a phase 2 SBIR grant, one single patient of a known drug. And the trial that LaPella did do, Jason Heffron was the PI of it. It was a multi-center dose escalation study. It was 13 subjects. So we can show that it's safe, which is number A1 with the FDA, and that there is a signal of efficacy. We can demonstrate, most importantly, we demonstrated that the whole blood tacrolimus really did not go up during that time and only very transiently increased, but all within the safety limit. And we've shown patient that has noticed significant improvement with decrease the sites of bleeding with the installation. And that's what a phase 2A study shows. But moving forward, it's going to get very complex and expensive because you need to do the double-blind placebo-controlled multi-center trial. Clinical trial is expensive. So, and then finishing up, I want to mention something that probably people wouldn't have thought of, but it was so critical for us. Building the ecosystem, stakeholders, and advocacy. The FDA talk about it all the time. And I got lucky that a man in Cleveland area who suffered from radiation hemorrhagic cystitis found me at Beaumont Corewell, and he told me he was suffering so much and there was nothing out there that he formed a 501c3, that a nonprofit radiation cystitis foundation, and him writing a letter to the FDA and to the SBIR reviewer that the stakeholders, the public is crying out, and him and his friends with the disease that they need. I think it went a long way. It was very helpful, way more helpful than 5 letters from chairmen of urology from across the country. That one patient, but a ecosystem stakeholder advocacy, and they helped build up a patient registry and such. This stuff is needed not only for a drug approval, but I believe it's needed very early from the stage 1. So I talked about 7 things. So first, move into clinical as soon as possible. Do your animal models, get your IP, and if it works, move on. And two, I do believe of thinking through a patent and not publishing your abstract like I did, almost did, or your paper before you file a patent is just critical. SBIR grant, the Art 43 and 44, believe is a good way to go, but you got to have a real company, somebody who's a CEO of a biotech whose CV can stand up to an SBIR review panel. Think about an orphan designation. Think about a 505(b)(2) reposition of a drug for a different indication. And think about patient advocacy. What's your disease? Do they have people that cares about? But because getting a letter from this type of matters so much with the SBIR and the FDA. And lastly, it's really, it takes a group of effort. I can't believe that I'm close to mentor, colleague, and pupil from both the University, Pittsburgh and Corewell. And I'm so grateful for them for helping me working on down this path together. So, but right from the date, look, remember my first slide, the Valley of Death. I was on the other side. I'm a clinician at heart. So to me, it's not about a paper or a grant. The idea is to help a patient to get better. I cross the valley of death to the bench side, and together we cross the back to move it into the clinical. And each one subsequent valley of death can be conquered with the team, and you just keep going at it. If you have a good idea and you can have a patent that you can move forward with. So, and I'm here really just talking today, and I'm so appreciative is that people has helped me cross these bridges, and I've learned that some lessons the hard way. So I'll just be happy to see if anybody that think that I can be helpful or run an idea, bounce something off. Please let me know. If there's any barriers that you've encountered in translating your research. Have you ever considered about patient advocacy group? If you have any questions with SBIR funding, please ask me any question now. Or if you're not comfortable, just email after this call. I'll be happy to, try my best. Thank you.